Before we get started, I have a quick favor. I've been self-funding the Finding Genius podcast for 5 years now. I've done over 3,000 episodes. And as you can see on YouTube, we're up over 1 million views on the channel, which is fantastic. The next thing I really want to push on is to get up to 10,000 subscribers. Because once we do, we'll be able to put a donate button and we'll be able to solicit donations to help keep the podcast running and to also get the Finding Genius Foundation moving along. We have a big project studying anxiety, depression, and PTSD and working on a product to help people overcome these problems because I've seen them explode recently after the last 2 years of the whole virus situation. So if you would, please subscribe to the podcast. That would help us tremendously. Give us a thumbs up and check in the description for Buy Me a Coffee. It's about $5. If you could buy me a coffee, I'd really appreciate it. It would help keep the channel going, and I love coffee. Thank you.
Forget frequently asked questions,
common sense, common knowledge, or Google.
How about advice from a real genius? 95% of people in any profession are good enough to be qualified and licensed. 5%
go above and beyond.
They become very good at what they do, but only
0.1%
are real geniuses. Richard Jacobs has made it his life's mission to find them for you. He hunts down and interviews geniuses in every field: sleep science, cancer, stem cells, ketogenic diets, and more. Here come the geniuses. This is the Finding Genius Podcast
with Richard Jacobs.
Hello, this is Richard Jacobs with the Finding Genius podcast. My guest today is Aristo Vojdani. He's a chief scientific advisor at Cyrex Labs. So they have looks like a new panel that measures reactivity to 180 food antigens that may come from cooked, raw, or modified food. I've taken, panels maybe that are somewhat similar to this to see what I'm allergic to, what my body likes, doesn't like. So looks to be very interesting. So welcome, Aristo. Thank you for coming.
My pleasure. Thank you so much.
Yeah, let's start with just a little bit of background. How did you come to work here at Cyrex, and what were you doing before?
Well, in 1979, I came to UCLA to do my postdoctoral studies in the field of immunology. During those 2 years, between 1979 to 1981, I had an opportunity, in addition to my research, participate in 2 different workshops, which really, when today I look back, those 2 workshops completely changed my life.
Oh, really? How so? What were they about?
Yeah, so in the Department of Immunology, they announced that there was a workshop about the new method called enzyme-linked immunosorbent assay, which is good for measuring antigen and antibody level. So then really I didn't have much money. I had to pay $350 to participate in that workshop. I did that. And today I'm saying that that was the best $350 ever I spent in my life.
Wow.
It was during this workshop, the instructor taught us how to take an antigen from bacteria, for example, and coat, like E. coli, coat a test tube with that antigen, add human blood or human serum to that, and after that, go through some steps of washing, and if the blood sample contain antibodies, that antibody will bind to the antigen in the test tube. Additional reagents resulted in color development, and the color development was proportional to the concentration of antibody in the patient's blood.
How sensitive was the test, by the way? Did it discern very low levels?
Well, you could differentiate between low level, medium level, and high level. Let's put it that way. So that was my experience about how to measure antibodies accurately using advanced method, which was developed in 1976 by 2 scientists in Germany who won equivalent of Nobel Prize of Germany for that method. And I learned that method through that workshop at UCLA. And today there are close to 18,000 different kits manufactured that they are using ELISA methodology for diagnosis of different disorders, including the one that we are dealing with, immune disorder.
So what are some examples of, what ELISA is good for figuring out in people? Is there early detection at all? And again, what conditions is it really good at discerning?
Again, let me give you two excellent examples. One of them was during HIV/AIDS. We're measuring antibodies by ELISA to detect antibodies in the blood of patients with possibility of suffering from HIV or AIDS. During COVID, exactly the same thing. We use that methodology, including we are using that today in our laboratory for measuring antibodies against SARS-CoV-2. It's a highly, highly reproducible, sensitive, and that's why there are so many kit manufacturers that are using this methodology for diagnosing of different diseases in the field of medicine.
Okay.
That was one. The second workshop was about a type of cell in our body called natural killer cell. And that time, we knew about that type of cell, and practically they showed us if we take tumor cell and add blood from healthy person to those tumor cell lines, observing under the microscope, that first contact created between the natural killer cells from our body, it's a kind of white blood cell, with the tumor cells, and 15 minutes later, we could observe that natural killer cell releases something. Today, we know that they're called granzymes or enzymes that can poke a hole in the membrane of the tumor cell and destroy the tumor cells in few minutes. And that's how I became interested really now, not only in humoral immune system. The first part about antibody is a component of the immune system called humoral immune system. The part about cell-mediated immunity, which I gave you example of natural killer cell killing tumor cell, it's called cell-mediated immunity. So today, actually started from 13 years ago at Cyrex Laboratories, we started using these two methodologies. One, enzyme-linked immunosorbent assay for measurements of antibodies against example of different foods. Based on antibodies, we can identify individuals whether or not they have allergies, hypersensitivities, or immune reactivity to different food antigens.
So do people produce antibodies to all food, or only things that they perceive as being foreign in a bad way? What's typical for a normal person that doesn't have a food allergy?
Our body is extremely, extremely smart. What happens that during the first 6 months of life, the body gets exposed to variety food antigens. Of course, first is milk, and after 6 months, we feed the babies with different food. Our body, or our body's immune system, learn how to live in harmony with food that we eat on daily basis. So when we consume food, the food has protein, right? Our digestive enzymes digest the proteins to peptides. We have another set of enzymes called peptidases, break those peptides to amino acids, and the amino acids get absorbed into the blood and give the fuel to our body. So under normal condition, we should not react to any food, and that because that food is getting digested. And also, we have a set of cells in the mucosal immune system which induce a mechanism called tolerance. So we have to tolerate food that we consume by a type of cells called regulatory T cell, which prevents immune reaction to food. So the food will be ingested and digested, and then will get into the body in a form of amino acid. But unfortunately, in some cases, some individuals may not be able to digest the food entirely. Or let me give you a classical example. If I consume peanut, my body learn how to digest peanut protein. But unfortunately, because in peanut butter, we have also aflatoxin, meaning toxins from the environment got into the proteins of peanuts. The digestive enzymes will not be able to digest entirely the peanut proteins. So some of those undigested ones will be taken up by cells of the immune system if the patient is having leaky gut, and then by reacting to them, produce different types of antibody. If they produce IgE antibody against the peanuts, we call that allergy or hypersensitivity. If they produce IgA or IgM or IgG antibodies, we cannot call that allergy, but we call that delayed food immune reactivity, which has different consequences. Some of them are autoimmune diseases because certain foods share significant homology with human tissue. So when the immune system making antibodies against, let's say, milk, and milk shares homology with islet cell antigen, antibody made against milk can react with islet cell antigen, and that individual may develop type 1 diabetes. So we have lots of examples of antibody that we produce against undigested food can turn around against our body tissues and induce autoimmunity. And by the way, you can see the journal of autoimmunity right behind me.
How do we, can you tell before you eat a food, are there ones that are universally allergy causing or problem causing, or does it totally depend upon the, the genetics and epigenetic state of the person eating them?
Yeah, depending on the person. For example, peanuts may induce real allergy in 5 to 10% of, children or adults, but that doesn't mean that each person will consume peanut proteins will develop type 1 allergy, which is IgE mediated. So we have to test for that, either by a skin test. The patient goes to the doctor, they inject the antigen into the skin, and within minutes, if the patient is making IgE antibodies, will form some kind of skin positive reaction in a kind of redness, inflammation. And based on that, the doctor determines if the patient is really having allergic reaction. The second possibility is in the laboratory, we take the blood of the patient using the same method I explained earlier that I learned at UCLA, and measure how much IgE antibody that individual is making against that specific food. And based on that, we determine whether or not individual is having allergy to that.
Okay, is 180 enough? Or do you need to cover 1,000? Or, are these the foods that are typically most problematic?
180 is a good example of most common foods that we consume. Okay, so my involvement in this field became more serious in about 2005. So I came with the idea that in 1921, the first article published in the literature claiming that some people can react to cooked food in addition to raw food, but nobody really paid attention to that. And then there were additional publications showing that some individuals reacting to modified food, let's take example, almond, raw versus, when we dump that to heat, exposing that to heat, right? Called roasted. So some people may react to raw almond, but not to roasted, but many may react to roasted peanut, almonds or peanuts or, nuts in general, but not to the raw. So the question I asked in 2005, if certain food, we eat them in a form of cooked, the best example, potatoes, why, or beans, why allergies using raw bean extract and doing skin testing with that?
Well, also, too, on some of these foods, I've had dinners, I'm sure you had too, where the potatoes were a bit undercooked or overcooked. So there's probably other foods too that tend to not be delivered, at dinner time optimally sometimes, let's say by bad cooks. And therefore they trigger allergenic responses when maybe they shouldn't.
Well, the point I'm trying to make is some people may react to raw food, let's say shrimp, but many more may react to cooked shrimp. And so therefore, in the laboratory like Cyrex, we are the only one based on the same idea which I came with in 2005, asking if we eat food in a form of raw or cooked, why we are measuring all the laboratories that I know. They are measuring antibodies only against raw food. We are not eating every food in the raw form. We are eating them some in the raw, some in a cooked, some in combination.
Yeah, what about the combinatorial effects of different foods that may influence each other?
So I talk about synergistic effect.
Yeah, like if I eat potatoes with something with a lot of coriander, I don't know, I'm just making it up. Would the two interact somehow to make it worse for me?
That is an excellent question. I'm saying that,
Hey, I got one.
yeah, let's first solve the issue of raw versus cooked.
Right.
Oil laboratories are measuring antibodies only against raw food, okay?
But why? Is it easier or what's the reason?
Because they don't have the knowledge. I developed the first food antibody testing in 1986. Some of these laboratories at that time, I was using only raw food, came to my laboratories and learned from me and went back and developed, actually copied to some degree what I was doing. But since then, they don't have the knowledge how to change that. The only laboratory that measuring antibodies against food, if we eat that in the form of raw, if we eat that in the form of cooked, measure antibody against cooked. Now, let me answer your question specifically. So someone published this article about 10, 15 years ago who took sausage versus ingredients of sausage. What are ingredients of sausage, Richard?
The meat itself, the casing, yeah.
Right, the casein and gluten and wheat.
The filler, yeah, if you put that in there, which you'll do.
So they took each one of those separate. They inject that to different individuals, skin testing. They did not react to wheat. They did not react to milk. They did not react to meat. So that's now. But when they isolated wheat or milk from the sausage and injected that, many individuals reacted to that. So what do you think? So isn't that some kind of answer to your question? When we mix several foods together, plus all the...
It might just be that there's more of a chance if you have a dinner meal that has like a lot of different components to it, you'd increase the chance that there's going to be an allergic reaction just because of all the different foods. But I don't know if that answer is if they're interacting or not,
Well, we know for sure that food additive, whether emulsifiers or colors, microplastics, lots of them, right? They bind, they form some kind of chemical reaction, like food coloring, emulsifier. They bind to the proteins, and now it became completely a new protein. We call that neoantigen, which is different from the original one. And therefore, they react to that. That's one mechanism. Second mechanism is some of these food additives, they change our gut microbiome. And change of gut microbiome results in leaky gut. And entry of some of these undigested food proteins into the blood, and then we react to them, and we make antibodies, which may result in allergies, hypersensitivities, and even autoimmune diseases. So there are several mechanisms involved in the induction of these inflammatory and immune reactivities, including allergies and hypersensitivity.
Okay. So what does that tell you about allergy testing using different foods? Do you now have a cooked versus raw assay? Do you have a mixed foods assay? Like what needs to happen then to accurately assess what's going on with people?
Yes, unfortunately, the laboratories who are using only raw food, they are...
what actually needs to be added too is very common, like cooking oils. There's so much food has these cooking oils, whether it's being cooked by a person or being made to be cooked and then reheated later. That seems like if you're going to do anything, if you're going to go into the cooked realm, you want to include that kind of stuff, because I wonder how cottonseed oil affects someone's allergy versus no oil. Yeah.
Yes. So at Cyrex, probably, again, we are the only laboratory also. We are measuring antibodies against oil. But you may say oil, it doesn't have protein. It has oil, right? It's oily material. But there is small amount of proteins in oil. We call them oleosin. And that's why at Cyrex we are measuring antibodies against these oleosins, which are components of the different oil.
Yeah, also high fructose corn syrup, I don't know. It's I can see how complicated it is, but have you found any reason why people are not using any cooked foods, and how many people's, allergenic assays are wrong because they're based only on raw foods?
Well, the only answer I have is really the laboratories do not have the knowledge how to prepare the extracts of food from raw versus cooked. What they do, they go to different companies who sell allergen, including food allergen. But the companies who sell these food allergens, they prepare them only from raw food. And so therefore, they can take those raw food antigens, put them in a test tube and measure antibody against them. But to be able to prepare allergen, antigen from cooked food, you have to spend time. It takes years of developing this. I spent between 3 to 5 years to develop these extracts for Cyrex Laboratories, and therefore Cyrex Laboratories today is the only one measuring antibodies in raw versus cooked food.
So what are you seeing when you provide assays? Are you doing a raw versus a cooked one for the same person? And what are you seeing? Is there a lot of overlap or a little overlap?
There is a lot of differences in their immune reactivity. Depends from one person to person. I'll give you example that this is in my article published in the journal Food 3 months ago. Let's take very simple raw egg versus cooked egg. I took 3 different individuals. One of them reacted to raw egg proteins.
Yeah.
Second one reacted to the cooked one, and third one did not react at all. So if we measure antibodies against only raw egg food proteins, we are missing 1/3. The third person who reacted to cooked food, in this case, cooked egg, will get false negative results, suffer from allergy, and nobody will find out what the patient is suffering from.
Okay.
So that's why the importance of what we do at Cyrex Laboratories.
Yeah, that's very interesting. Is this, if I go to my doctor and say, hey, doc, I want to get an allergy test, using this assay or that one, is Cyrex right up there? Is it recommended, or is it still, you have to know about it and ask your doctor?
You have to know about it because Cyrex is only one laboratory out of so many in the country. And again, the doctors like to do skin testing, the allergy. That is their method of choice.
Are you actively trying to license this to as many clinics as you can, or how do you get the word out about it?
Well, again, it takes really a lot of marketing and how you present yourself to the whole world. It takes some time.
How long has this assay been in existence now?
At least 12 years.
Oh, all right. Well, they need to hurry up. Huh. What kind of push?
Richard, that's why we need people like you. That's what this interview.
You just say, what's cooking? It's funny you should ask this assay. So what kind of pushback do you get when you presented this to labs and to, insurance plans and all that?
Well, these laboratories who are doing this, they're not billing insurance. If they will bill insurance, they will reject their claim because we are not using the classical methodologies. We are using new methodologies, which they did not approve.
That's very foolish. It'll be interesting. There's so many things you could experiment with. Let's say someone has allergies and they're a smoker and they stop smoking. Now you'd get a whole cascade of epigenetic change. And how would that manifest and what they're allergic to or not, Okay.
Let me add to that also the following, that someone is consuming a lot of sugar, whether it's corn syrup or whatever, or cigarette smoke. Okay, you gave a good example also. So what happens that cigarette smoke has a capacity, if we'll get into the digestive tract or come in contact with food indirectly, right? Like passive smoke, right? The food smoke, the cigarette smoke, the chemicals will bind to the protein.
Right.
And I call that neoantigen. That's one way. Second way is many sugars, when get exposed to heat in the presence of protein, so we eat a lot of, let's say, lots of food these days contain sugar and protein. And when exposed that to heat, the sugar bind to the protein, forming new antigen called advanced glycation end product.
You've heard of that, yeah.
I'm sure you heard about that. So advanced glycation end product can get into different tissues, causing inflammation, tissue damage, in particular kidney damage, cardiovascular disease. And because we have receptor for advanced glycation end products in different tissues, including in our brain, now these advanced glycation end products from food can damage the blood-brain barriers, get into the brain, causing inflammation in the brain, which can or may result in neurodegenerative disorders such as Alzheimer's disease. So in my article published in 2009, not only I showed that individuals can react to modified food, but the same individuals who reacted to modified food also had much higher levels of advanced glycation end product, oxidized LDL antibodies. So all of that shows that modified food is a major contributors of inflammation, autoimmunities, and neurodegenerative disorders.
Yeah, I'm sure gut microbiome affects us tremendously. What someone's allergic to versus not. I guess if you don't have the bacteria to process a certain kind of protein on food, then you could have an allergy. But if your gut microbiome changes, maybe that would make it go away or get worse. There's a lot of factors.
Yes, Richard, in scientific journal, first of all, the word microbiome was introduced in 2001. Since 2001, 150,000 manuscripts with the word microbiome in their title or in the abstract are published in medical journal. That shows how important is our gut micro and microbiome. And that's why everybody in the field of microbiome paying attention to the bacteriome part of the microbiome. So very recently, about 2 years ago, I again said that why everybody is looking at the gut microbiome? How about the mycobiome, which is the yeast?
So they had very few people, or the virome.
Yeah, and then we have the virome. So that's what I added measurements of antibodies, not only against the bacteriome, like E. coli, Salmonella, Yersinia enterocolitica, Clostridium difficile, other bacteria, which we use to measure antibody against them. So very recently, we added measurements of antibodies against yeast, such as Candida albicans, tropicalis, and others. I'm sure you know that in many hospitals, immunodeficient patients, cancer patients, after chemotherapy, they really die due to fungal infection, especially subspecies of Candida called Candida auris. So that's why we measure antibodies against different species of Candida albicans. And because yeast that we consume in our food are kind of cousins of Candida, they share homology. Also, we measure antibody against that. We know that patients with Crohn's disease make antibodies against yeast, make antibodies against Candida albicans because of antigenic similarity between these, and also food-derived mold. For example, cheesy food get contaminated with mold. Also, we isolated those molds from food and we measure antibody against them. So we measure antibodies against the yeast, fungi, the molds, the aerobic bacteria, anaerobic bacteria. The neutrophils' job in our body is to fight against our microbiome when they are in an imbalanced state. And so if neutrophils will win the battle, everything will be okay. But sometimes, unfortunately, the yeast and the bacteria are killing the neutrophil. And the neutrophil release all kind of factors called neutrophil activation factors. And then we react to them, and that can also destroy our tissue, resulting in autoimmunity. So we do that as well. And so we came out with this new panel called the Biome Burden Panel, which includes the bacteriome, the microbiome, the yeast, molds, their virulence factors, and all those components which cross-react with human tissue. So therefore, this is some kind of addition and completion of something was missing by looking only at bacterial antibodies, looking only at the bacterium part of the microbiome, and not looking at the microbiome of the microbiome.
Boy, is it complicated? Yeah. I guess a real assessment would be that this is a good way to look for, the presence of various yeasts and then fungi and all that, in various situations. So it's be very, very useful.
Yes. So that's really comprehensive way of looking at the picture. If you look only at half, you are missing the other half, like raw versus cooked food. you're missing half.
But what about has anyone done a study where people have a particular kind of cancer and the diet is highly controlled, and you look for the a change in allergy profile? Or I would guess if you administer chemo, the allergy profile probably radically changes. I bet it causes cascades and cascades of change, and then you'd see very different results from this assay.
Yeah, we know for sure that gut microbiome plays a significant role in cancer treatment, whether it is chemotherapy or other therapy. So if you put the patient on the right diet and the right bacteria survive in the gut, that healthy gut bacteria help the patient to overcome almost all the side effects of chemotherapy. So diet is a major component of cancer therapy. Unfortunately, many doctors do not pay attention to that. But recently, I have seen several oncologists emphasizing that that's why they hired dietitians to help cancer patients, which is a very good step, because the microbiome is the one that we should keep healthy in order to take care of the body and protect us against damage done by cancer cells.
So you've been pushing on this for 12, 13 years. Are you getting positive responses from at least functional medicine people or pathologists or who's open to this, who's not?
Functional medicine people, absolute. But unfortunately, doctors, practitioners really do not have time to read about some of these publications that I publish or other individuals are publishing. They only are influenced by the sales people of the laboratories. Go to their clinic, and those who have a better marketing skill, they convince the doctors to use their methodology. They claim, for example, their method is the best method in the world. But really, Richard, all the methodologies developed in the field of medicine are good. What antigens, what proteins, how pure are these proteins you are using in your assays makes huge difference. Unfortunately, only small percentage of functional medicine doctors are familiar what we do at Cyrex Laboratory.
So how much does an assay cost? If I were to get it, I'd have to go to the right doctor and tell, all right, you got to go to Cyrex Laboratories unless they knew it themselves. And then how much is this assay versus the current raw one? And do you package in raw and cooked for, should you do both? I guess through your lab, you ideally should do both, raw and cooked, right? But if we do anything, at least cook.
So your question is really very, very good question that many laboratories, again, they go to the doctor and say, our method is good method, very good method. Talk about only the methods. They don't talk about raw versus cooked food. And they say our pricing are half a price of Cyrex Labs. And they get convinced because, cheaper for the patient, they think it's better. But they forget that at the same time, they are getting false positive and false negative. So if you want reliable test results without false negatives and without false positives, then you have to pay a little bit more. If you want to drive, I don't know, Hyundai, or you want to drive Mercedes or, another.
I'm with you.
Car, you have to pay for it.
Yeah. Well, I mean, do you, of the functional doctors that you've dealt with, you should have a lot of clinical outcomes gathered from the past 13 years. Where is this packing the most punch? Where is this the most beneficial when used by functional medicine doctors? Has it kept people alive that have cancer? Has it healed or stopped or put into remission any autoimmune conditions? Where do you see that, again, clinically it's having the most effect?
I think lecture, presentation. We go to different meetings, but I'm only one person. How many lectures give every year?
Well, what about using the, the results? How many, I don't know, let's say you've dealt with 1,000 functional medicine doctors over time. What about reaching out to them and hey, you did our assay. What can you share about the patients that did the assay and then changed their diet or, eliminated some food? I think there'll be a good, cheap back door for you to, the space of things that you need to look into is so huge and so expensive that I would figure you'd have to do that to even get a glimpse of what's in there,
Yeah, definitely. One credit I can give to myself, not to being selfish, is exactly what you said, that I love to communicate with the doctor, saying that, doctor, you ordered food sensitivity and your patient reacted to 100 out of 180 food. At the same time, we did also lymphocyte subpopulation, looking at the cellular immunity of the same patient, and is having these abnormalities, this or that abnormality. So I called that doctor and trying to connect the dots. In that particular case, I learned from the doctor and the doctor learned from me. And those are the doctors really benefit significantly from what offered at Cyrex Laboratories. And we have many of them. So we are doing that one by one.
I know that you're not allowed to say, or you got to be careful with what you say, but what can you say? Like, what trends or cool surprises have you seen from, it's anecdotal, the clinical data?
Cool surprises from what? From the doctors?
Yeah, from the doctors disclosing to you, whatever they can. I had 3 patients that did this assay, and we were able to help, 2 of them with XYZ because of the assay. This is great. Thank you. We're going to order more. I'm sure there's functional doctors that
a lot. We have that kind of response a lot. But let me give you another example that my laboratory is immunosciences lab also right here. That we do measure antibodies against viruses like Epstein-Barr virus, cytomegalovirus, herpes type 6, SARS-CoV-2, and other, as well as we do tests for Lyme disease. Okay, so a doctor send blood to me and send blood to another laboratory who is known to doing shortcuts in assay development. So 2 weeks later, the doctor is calling me saying that, Aristo, how come the test for Lyme is positive in that laboratory, but negative for viruses? And your laboratory exactly reported opposite, positive for viruses, but negative for Lyme. So I said, doctor, I'm ready to put my 50 years of reputation behind the test results reported by my laboratory. And I am assuring the accuracy, reproducibility of our tests because we use this material, we use that material, we use this method, that method, and also blindly every 3 months we get checked by the College of American Pathology because we are part of the College of American Pathology. And then you can look at our score. Our score for these tests are 100%. Why don't you ask the other laboratory who reported opposite results? So I left it there because I don't want to get involved in the politics of fighting between different laboratories. I can only describe my test results and stand firmly behind our laboratory test results.
Okay. So, all right. Yeah. I just, I don't know. I just wish that this was more available. Yeah. It just seems like I didn't know that. Like I said, I did an allergy assay. I didn't hear one word about the foods being cooked or not. So I have no idea if it's, I thought some of it was relevant, but maybe not. Interesting. So how can people get in touch and get this help and the proper assay?
Yeah, just really go on cyrexlab.com and find information for every test that we developed in the past 13, 14 years. We have 3 different sets of documents. One is called Clinical Application Guide, like 50 to 100 pages. Second one is Clinical Interpretation Guide, which explain the meaning of the test. And thirdly, Patient's Education, which is like 3 to 5 pages about each test. So please go online and look at the menu of Cyrex Laboratories, and hopefully you will choose Cyrex Lab as the laboratory of choice for measuring different antibodies, whether it is against food, raw versus cooked, against bacteria, viruses, parasites, yeasts, looking at leaky gut, looking at environmental factors at the cause of inflammation and autoimmunity. So please consult with, Cyrex, go online, cyrexlab.com, and find the information and consult with all those documents as well.
Okay, very good. Well, yeah, I wish we had more time, but it's been great to speak to you, Aristo. Sorry.
My episode. So, Richard.
Yeah. No, I encourage people to check out Cyrex Labs and obviously speak to your doctor and see if the assay is right for you or not. But thank you very much for coming.
My pleasure. Thank you. Have a nice weekend.
If you like this podcast, please click the link in the description to subscribe and review us on iTunes.
You've been listening to the Finding Genius podcast with Richard Jacobs. If you like what you hear, be sure to review and subscribe to the Finding Genius podcast on iTunes or wherever you listen to podcasts. And want to be smarter than everybody else? Become a premium member at findinggeniuspodcast.com. This podcast is for information only. No advice of any kind is being given. Any action you take or don't take as a result of listening is your sole responsibility. Consult professionals when advice is needed.